Thank you very much for that kind introduction. These are two very tough acts to follow. I'd like for you to join me in thanking Michael and Allison and Maddie, who's the really tough act to follow, for doing everything humanly impossible to advance research, not just for Maddie, but for everybody born with PA. So thank you very much. So I've been asked to give you a brief illustration using a single therapy development journey of how all of us, Congress, patient advocates like yourselves, academic investigators, industry, NIH, and the FDA are all working very hard together, and how we need to work even harder, even faster to save these lives and these capabilities for those of us living with rare diseases. The case in point that we use to illustrate all that is a single ongoing therapy development, a protein therapy for Friedreich's ataxia. And so just to set the stage, Friedreich's ataxia is a rare, multiple system, progressively degenerative, recessively inherited genetic disease. And what does it do? It deprives our patients of much of their capabilities over time of balance and coordination, of speech, hearing, vision, causes diabetes, and scoliosis or curvature of the spine, a great deal of fatigue, and a very life-threatening heart condition. There's no cure, obviously. We do have one approved treatment that slows the progression of this disease, and which makes time of the essence. Time is important to us because we need to not only continue to slow, but to stop and reverse, and we even need to prevent these systems before they occur. So time for us is brain, time is muscle, time is life itself. That's why we must all proceed together with such urgencies. To do all of that, we need a lot more research funding at the NIH. We need a lot more regulatory flexibility, so we need funding and staff at the NIH and the FDA. To further set the stage, let me tell you how FA is caused. It's caused by low levels of the frataxin protein, a protein that is absolutely essential to life and is responsible for energy production in all of our cells. The problem is in the mutation that causes Friedreich's ataxia, there's an expansion of part of the genetic code on the FA gene. You can see in this diagram how in the top part of the left, you have the normal expansion of that genetic code, and you produce a whole lot of frataxin protein. You produce a full load, 100% load. In the bottom graphic, you've got an expansion of that genetic code at that one location on the frataxin gene, which leads to production of a much smaller, about a 15% of normal load of frataxin protein. There's two pieces of really good news here. You can, that small amount of frataxin protein at the bottom is not toxic. We have no abnormal or toxic protein processed by this genetic mutation. What is processed is perfect frataxin protein. So we don't have an abnormal or toxic protein to have to eradicate. Second piece of good news is the parents of these FA patients, like a couple right here, they don't have a full load of frataxin protein either. They've got about 50% of frataxin protein, and you can see they're beautiful and very healthy. And so, like my wife. And so we don't have to get our patients from 0 to 100%. If we can get them from their normal, what they're getting now, about 15% of the frataxin protein, up to 35 or 45% of the load, we expect to be profoundly beneficial. We expect to be beginning to spell the four-letter C word, and you know what that word is. So I will say that that FARA was established in 1998 to support the kind of research necessary to advance those two causes. The most important, the most promising beneficial therapies, therefore, because of that genetic mutation, are to either fix or supplement the gene, or to supplement the frataxin protein directly. And FARA was established to advance research in both of those strategies. So the illustrative example of an ongoing therapy development I'll use to illustrate how we're all working together and need to work together even faster and harder begins in 2001 when I got a call from a cardiologist at Wake Forest University in Winston-Salem, North Carolina. He called to tell me that he had been working with other scientists at Wake Forest on a strategy to deliver a protein in a different disease to the mitochondria, the energy-producing factories of all of our cells. And he decided to go online and try to find a rare disease that was described by mitochondrial dysfunction that this protein delivery mechanism might be useful in. He found the FARA website, on which we describe Friedreich's ataxia as a mitochondrial dysfunction disorder. So he called me. He said, I wonder if this program might work for Friedreich's ataxia. I drove to Winston-Salem, North Carolina that weekend, sat down with Mark in his lab, met with one of his patients. He described that process, and I said, please apply to FARA for a grant. He did so. 6 weeks later, he got that grant. Only about a $30,000 grant, but it was accompanied by a mating pair of the mouse model we got from our French lab in France. So he used that grant and that mating pair of mice to start building his mouse colony and trying that process in those mice. I can barely read this. In 2005, he, based on the preliminary data he gathered from that $30,000 grant in his mice, he was ready to apply to the NIH for an R21 grant. It's a small, exploratory, high-risk, high-reward grant, but a lot larger than our $30,000 grant. And he sent it to the National Institute of Neurological Disorders and Stroke, NINDS. At the time, I happened to be very fortunate to be serving on the National Advisory Council of the Neurological Institute and was able to participate in the discussion of that grant and recommend unanimous approval of Mark's R21 grant, which was granted. And on the basis of that larger grant, over the next few years, he was able to establish the use of that protein delivery mechanism in his mouse model that we had provided him. Then, only a few years later, 2013, on the basis of those data, he formed a company. He called it Chondrial Therapeutics. And then the next year, a FARA board member assembled some angel funding that permitted him to establish the preliminary proof of principle in the mouse of both manufacture and delivery to mitochondria in the mice. Then in 2014, he received funding from NCATS, the National Center for Advancing Translational Sciences, as part of one of their therapeutics for rare and neglected diseases. His application to that was accompanied by a FARA letter of support. And so there again, I was able to follow the progress of that application because I was then serving on the National Advisory Council of NCATS. So I would encourage all of you, all of the patient advocates, to consider having a representative participate in the public sessions, at least, of those National Advisory Councils. All 27 institutes and centers have a National Advisory Council that meets 3 times a year. You can learn a lot about what they're doing for all of us. You can begin to meet your program officers and so forth. I encourage you all to take a look at that. They're public sessions of those councils. Then in 2016, Chondrial Therapeutics garnered some additional venture capital that permitted them to go forward. And in 2019, they participated in what's called a reverse merger, which rebranded Chondrial Therapeutics into Larimar Therapeutics. And also in 2019, marvelously, they began their phase 1 clinical trial. This was after several iterations of FDA meetings to which Larimar, and before them Condrial, invited FARA and FA patients to participate. That's something else all of you can do. Be invited by your industry partners to go to their FDA meetings, represent the patient's voice at their meeting. Because the FDA can't invite you to those. It's an industry partner meeting. Then in 2002, great news, they started 2020, they started their phase 2 clinical trial, again, after several iterations of FDA meetings in which they included FARA leadership and FA patients. Then in 2024, they opened and are still conducting a pediatric study and an open label extension. Then I'll say just 2 days ago, now try not to get too emotional about this, just 2 days ago, the FDA announced the award to Larimar Therapeutics for this program, what's called Breakthrough Therapy Designation. This is a wonderful program that's granted to a therapeutic approach that is expected to be profoundly more beneficial than anything else available to that set of patients. And it also is all hands on deck. Everybody at the FDA is on call to help expedite the review of that therapeutic. The FDA had already awarded to Larimar Therapeutics what's called the START program. And the START program gives you a lot more frequent and less formal communications. That combination of urgency and flexibility expressed by the FDA gives Larimar a great deal more confidence that they can successfully submit this summer their biologic license application for approval. This summer, in June of this year. That expression of expression of flexibility and urgency on the part of the agency also gives everybody in the FA community, everybody in the rare disease community, that much more hope and confidence that the right thing will be done to save the lives and capabilities of our patients. It also gives all of us additional commitment to continue to advocate for the resources and the staff needed at both the NIH and the FDA to continue doing the work that we all need to succeed and to save the lives and capabilities of all of our patients. So this is a wonderful story. I'm really hopeful about getting our second or third approved treatment, but you can see from this graphic, it took us 25 years. That's too slow. We need to greatly accelerate that process. We need to continue working all together, everybody in this room, everybody in the Congress, everybody at the NIH, everybody at the FDA. We need to work harder and faster together to make this happen much more quickly. Thank you very much.